Hello, so my name is Darlisha D'Ampert, and I will be discussing interferons and superantigens and what they are pretty much and the clinical significance of them both. This is for exam 1D discussion. So I want to start off with super antigens. Pretty much what they are, they're just overactivations of T cells. These proteins, they're capable of eliciting a strong response because they activate more T cells than a normal immune response. So, which means they produce many viruses and bacteria that interact with the TCRs. However, super antigens, they are activated T cells and they may produce systematic diseases, and they characterized by systematic inflammatory reactions. So, just kind of an example of this, I found this on the U.S. National Library of Medicine, National Institute of Antigens, National Institute of Health. Pretty much they talked about the unique features of super antigens and how they bypass the antigens processing the mechanism and specifically binds to TCR V segment and forms the trimolecular complexes. And along with this is major histocompatibility complex class 2. There's a lot that goes in with it. They are implicated in pathogens of diseases, bacterial, viral, and even fungal origin. So they're also known to play a role in the autoimmune diseases. They're like therapy for the super antigens, and it's being worked upon. It's still in the process. Some studies have shown that the gamma globulin are containing specific antibodies to these super antigens, and they show the activation of these T cells and also the cytokine production by them, and in result they form a dramatic recovery from super antigen mediated diseases. So these are super antigens in the therapy of cancer, and it's being widely explored, as we know. So it's a hot topic in science. Next, I would like to talk about are the interferons, and just a brief overview of it and what I got from it. They're proteins that are produced by certain viral infected cells, and they're known to be warning messages for these cells. They work on uninfected cells and stimulate antiviral proteins. There are certain interferons that inhibit tumor formation, such as, hopefully I'm pronouncing this right, but Kaposi sarcoma, HIV, hepatitis, and there's several others, but those are just those to name a few. And just to point out some of the clinical uses of interferons, and I got this from the U.S. National Library of Medicine, National Institutes of Health. One example is the chronic HBV infection. It's one of the major one of the major antiviral applications of interferons. They It's a heterotrophic DNA virus, and similar to other diseases, HBV infection is associated with contaminated blood or blood products. But it's not a sexually transmitted disease. Well, it is. It's commonly a sexually transmitted disease. Excuse me. There is an effective vaccine for HBV, and studies have shown that it greatly decreases incidence of this disease. However, there's a large number of those chronically infected remains across the world. So there are still some concerns about it. At present, the chronic infections with HBV are treated with a combination of PEG interferons and one of the several nucleotide analogs that inhibit the replication of HBV are genome, and some of those being like tenofovir. So that's just one to name a few. A little bit to touch on it just real quick. There are more effective anti-HBV nucleotide analogs. They're under development as we speak of, but with PEG interferons therapy, approximately according to their statistics at the National Library of Medicine, National Institutes of Health, about 60% of patients demonstrate a significant anti-HBV response. So, in other words, it's more effective than therapy with any other treatment alone. So, that's just an example of what's going on in medicine that correlates to these, to a lot of the stuff that we're studying, such as the superantigens and the interferons. But that's pretty much what I got out of it, the gist that I got out of it. Please do not use this video, but that is it. Thank you.